Hello everyone, and welcome to this podcast on vesicular transport and the secretory pathway. So let's begin with the nucleus. Now, protein doesn't start off in the nucleus in the secretory pathway, but it's important to draw it because the nucleus is continuous with the first organelle of the secretory pathway, and that is the ER, the endoplasmic reticulum. The next compartment of the secretory pathway is the Golgi apparatus. And the Golgi apparatus is a system here of stacked membrane sacs. And we'll talk more about these when we get to that part of the secretory pathway. So let's draw the plasma membrane over here. I'm just going to put PM for plasma membrane. And so a protein that needs to go from the ER, I'll just make a little dot in there for the protein, will fuse with the Golgi, and then the Golgi will move that protein in vesicles all the way through the Golgi. And it'll go through each of these stacks. And then the vesicle will butt off of this last part of the Golgi called the trans-Golgi, and we'll come to that later. But then we'll have this vesicle here. And it'll contain our transported protein. It will then move to the plasma membrane, where it will fuse with the plasma membrane, thereby releasing the secreted protein. So what I've just described for you is the secretory pathway, also known as exocytosis. In addition to exocytosis, I should take a moment to describe the reverse pathway, so to speak, called endocytosis. In endocytosis, we have some kind of cargo, a protein of some sort maybe, or it could be a larger structure like a bacterium or a virus. And it's going to enter into the cell. And as it enters into the cell, it will enter in through a vesicle, which we'll call an endosome. We'll call it an early endosome. It will then fuse with a vesicle coming from the Golgi, which probably has proteins in it, certainly would, and it will become a late endosome. This will ultimately mature into a lysosome. And again, we'll talk about this whole pathway later, but in this podcast and the next podcast, we're going to focus on vesicular transport and the secretory pathway. And now I'm going to spend some time talking about, in more detail, vesicular transport. And to begin that discussion, let's talk about the vesicles. We have vesicles that move from the ER to the Golgi, between the different parts of the Golgi, to the plasma membrane, as well as in the endocytic pathway. So these transport vesicles must do two things. They have to have the correct cargo. The cargo could be proteins, it could be a specific kind of lipid, but whatever it is, it has to have it in the right vesicle. Second, the vesicle has to fuse to the correct target. We don't want a protein destined for the plasma membrane to move to a late endosome, or a protein that's supposed to stay in the Golgi go to the plasma membrane. So it's important that you have the correct cargo and it's transported to the correct target membranes. This, by and large, both of these are controlled by proteins on the surface of each vesicle. So let's introduce coated vesicles. As you have this membrane here, with the vesicle coming off of it, the vesicle itself doesn't just magically form. It forms because various coat proteins bind to the membrane and pinch that membrane out a little bit to ultimately form a coated vesicle. Then what will happen is these coat proteins will be lost, and you'll end up with what is referred to as a naked vesicle. And the coat proteins are free to go back and repeat the process again. So coat proteins, or coated vesicles, have two main functions. First, they help shape the membrane into a vesicle. Second, they capture specific cargo that has a specific destination. So each of these steps here, though it wasn't drawn, will have coat proteins bind to the vesicles and move them to the next target, all the way through this process. Now I would like to spend some time talking about a very specific coat protein, the one that we know the most about, and it's called Clathrin. So let's talk about Clathrin coated vesicles. Clathrin coated vesicles are known to form here from the Golgi, as it forms this vesicle that goes to the plasma membrane, and they're also known to form here during endocytosis, as vesicles form to move into the cell. So let's talk a little bit more specifically about how these Clathrin coated vesicles form. And to do so, I'm going to draw a membrane down here, and it's going to be the plasma membrane, so this will be the extracellular space, and here will be the cytosol. Even though I'm not talking about endocytosis for a few days, we're going to use this example of Clathrin coated pits and vesicles forming here because we know more about it during endocytosis. So during endocytosis, there will be various cargo proteins or molecules of some sort that need to get inside of the cell. And the first step that occurs is they bind to these receptors. And let's make a little key over here. The red dot there is the cargo. This is the receptor. And we'll add to this as we move forward. As soon as this receptor is bound with the cargo, a very specific protein will bind here. And this protein is called adaptin. There are many kinds of adaptins inside of a cell that will very specifically bind to specific receptors that bind to specific cargoes. The same is true of receptors. There are many kinds of receptors in the cell. Once the adaptin binds to the receptor, it will recruit the Clathrin protein. And then what happens is the membrane will become a little distorted, primarily because you're bringing in a bunch of proteins right here, and it starts to pinch that membrane up a little bit. And what we might see here now are more receptors with bound cargo. And then, of course, more adaptins with more Clathrin. And then at some point, a very short time later, it doesn't take too long for this to happen, the membrane becomes even more distorted, forming what looks like a stalk. Oh, and I should have mentioned that what's forming here, we call this the coated pit, because it forms what looks like a little pit here. What's forming here now is the formation of the Clathrin coat. Because what you'll have here, and I'm just going to draw a few just for simplicity, I'll draw 4, but you may have several more of these receptors here, each bound with that specific cargo that's trying to get inside the cell. And each of these has, of course, the adaptin and then the Clathrin. And then a very cool protein comes along. It's not my favorite protein, but it's certainly in my top 10. And it wraps around this stalk. In fact, it begins to wrap around the stalk in a way to help form it. So it comes in play much sooner than I drew it here. But this protein is called dynamin. And what it will do, in addition to forming this stalk here, it will facilitate the fission of this membrane, the dividing of this membrane. Pinch together these membranes in such a way that the vesicle will be released, still with the receptors and still with the cargo added in it. Of course, we would defeat the purpose if we lost the cargo at this point. And still with our adaptins added here and our Clathrin coat proteins. And if you look at an EM image of this, it looks like this cage that wraps around this vesicle. They're pretty cool pictures because it's there to protect the cargo and to form this vesicle. Now the next stage that occurs is the uncoating stage, removing the Clathrin coat proteins as well as the adaptin proteins to form this released naked vesicle. Carrying the cargo inside of it. This naked vesicle now is free to fuse with its target membrane. One thing I forgot to mention here, and that is that this dynamin is a GTPase, because this requires considerable amount of mechanical work here to push these membranes together to cause them to divide, and so it requires the hydrolysis of GTP. Now there are other coat proteins involved in other steps of this pathway of secretion, and they are called cope proteins, and they're involved in ER to Golgi and in between the Golgi stacks as shown here. Now I want to say a couple things about adaptins, and I'm going to say it on this slide because this is where I've drawn them. So I'm going to just make the notes over here. And adaptins are all the proteins in this process, of course, are important, but adaptins, they serve a couple crucial roles. They secure the clathrin, which is essential in forming this coat and eventually in forming this pit, and then eventually this coat, and then eventually this vesicle here. But the main point I want to make about adaptins and the role is that they're very specific. Remember, we said that these vesicles, one of the problems they have to overcome is selecting the right cargo. And it's the adaptins that do this because they bind to specific receptors that have specific targets as well. But if the adaptins weren't specific and they just bound any receptor, then they could bring in the wrong molecule. It also helps specify which part of the secretory pathway we're working on or which part of endocytosis. So the adaptins are very specific to where they bind. Now, next I want to talk about what we're going to do with this naked vesicle. Where is it going to go and how is it going to get there? So the goal is to get this vesicle to its target membrane, where it will fuse with the target membrane and release its cargo. But how does it do that? Well, it does that by and large by binding to the cytoskeleton, and we'll talk more about this later in the semester. And as it binds to the cytoskeleton, it will move along the cytoskeleton to get to its target. There are many proteins involved in this movement, and again, we'll talk about that later. But how does it know which target to bind to? So for instance, you wouldn't want this vesicle here that's destined to be secreted to fuse with a late endosome. There's got to be a specific way that it can reach its target membrane. So this here will be our target membrane. Doesn't matter which one we're talking about in this case. It's our target membrane. On this side of our membrane is our cytosol. So let's now draw our naked vesicle that we had on the last slide with its cargo in it. Now there are going to be multiple receptors and cargo in here, but I'm going to simplify it by just having one in here for now, because we have to add a couple things to this. And so once again, we'll have our key over here. So again, that's our cargo. This is our receptor. And now we have a couple new players to add here. One will be a Rab protein, and it's going to bind right here to the cell. And then we need something called a V-snare, which we'll put right like so. Okay, now we have something else that we're going to call the tethering protein. And it's going to be stuck in the membrane like so. And what it will do is it will bind to the Rab. So let's draw this vesicle getting closer here. And again, it's moving along the cytoskeletal component, and this tethering protein will wrap around it. We still have our V-snare and our receptor and the target. And I don't think I mentioned before, but V-snare, the V stands for vesicle, so we're going to find that on the vesicle. Now what happens is this tethering protein will move the vesicle closer to the membrane, so it can interact with one more protein we haven't mentioned yet, and that is our T snare. Structurally, it's very similar to the V snare, and we say T because it's on the target membrane. And so it will bring the vesicle down so that the V snare and the T snare can now interact. So let's draw that. And it interacts in such a way that they kind of wrap around each other. Now the tethering protein is still attached to the rab. And then what happens, and this is a really pretty fascinating thing, is that these snares will continue to wrap around each other, almost like a cranking system, until it brings this membrane of this vesicle closer and closer to the target membrane. So these will continue to wrap and wrap around each other until this vesicle membrane is about 1.5 nanometers from the target membrane. And when that happens, the vesicle will fuse with the target membrane. It will release its cargo. The receptors are still in here, but they just released all the cargo. Then, once the cargo is released, the V snares and T snares and the tethering proteins in the RABs are now recycled to redo this whole process one more time, or many times. So what we've described in the past couple whiteboards is how a vesicle is formed from the originating membrane using a Clathrin coat or some other coat protein, and then how that vesicle moves along the cytoskeleton and ultimately interacts with the target membrane and fuses with it, releasing the cargo. And this is what happens at each of these steps as the vesicle moves from one compartment to the next compartment to the next compartment. And it does so in a very specific manner. The specificity that we see comes in a few ways. Remember, we said the adaptins, but also these RAB proteins, RAB, and the tethers. These all add to the specificity, so the right cargo gets into the right vesicle, and the right vesicle fuses with the correct target membrane. Next, in the next podcast, what we will do is we're going to focus more on the secretory pathway specifically. But that's all I have for this podcast. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.